Hi everyone, I'm Chen Zixia from CSBL. Today my presentation title for the journal club is Poria cocos, water-soluble polysaccharide, modulates anxiety-like behavior induced by sleep deprivation by regulating the gut dysbiosis, metabolic disorders, and TNF-α NF-kappa B signaling pathway, which published on Food and Function at 2022. Poria cocos belongs to the Polyporoaceae family and grows on the roots of pinus species, with the best collection period being from July to September. It is commonly applied to treat multiple symptoms in the clinic, including palpitations, loss of appetite, mental anxiety, and insomnia. The chemical constituents of Poria cocos mainly include triterpenes, polysaccharides, and steroids. The main components in the aqueous extract of Poria cocos are water-soluble polysaccharides. Sleep deprivation can lead to many negative consequences, such as inflammation, intestinal barrier destruction, intestinal flora disorders, decreased attention, memory loss, behavior change, and so on. Studies have shown that sleep deprivation can trigger or further exacerbate anxiety disorders. A plethora of neurotransmitter and neuromodulator systems orchestrate sleep and emotional regulation, including norepinephrine, GABA, dopamine, glutamate, serotonin, and adenosine, as established in animal models or pharmacology works. So this paper uses sleep-deprived mice to create a model of anxiety-like behavior. The picture shows the downstream signaling pathway of TNF-α. The tumor necrosis factor alpha mediates its action through TNF-α receptor 1 and TNF-alpha receptor 2. The TNF-R1 and TNF-R2 are members of the TNF superfamily and share a cysteine-rich extracellular domain. TNF-R2 is expressed only on endothelial and immune cells, whereas TNF-R1 is commonly expressed in all cell types. TNF-R1 has a broader role in NF-kappa B activation than TNF-R2. TNF-R1 induces receptor trimerization and recruits the adaptor protein TNF-R1 related structural domain of DAS protein, which bind to a specific DAS domain in the TNF-R1 cytoplasmic domain. The TNF-R1 associated DAS domain protein also recruit TNF receptor associated factor and activates I-kappa B kinase via receptor interaction protein. The RIP1 is essential for TNF-alpha induced IKK and NF-kappa B activation. Activation of I-kappa B kinase leads to a phosphorylation, cubitination, and then degradation of the I-kappa B protein. The degradation of I-kappa B releases the transcription factor NF-kappa B, which translocates to the nucleus, binds to the kappa B site, and activates gene transcription. Upon exposure to the TNF-alpha, NF-kappa B is activated and leads to the expression of multiple inflammation-related genes. Subsequent activation of NF-kappa B is associated with tumorogenesis. This is the graphical abstract of the article. Water-soluble polysaccharides are the main components of Poria cocos. Rats were placed in a water tank with multiple platforms for 21 days to induce chronic sleep deprivation. The role and mechanism of PCWP in CSD-induced anxiety in rats were investigated by determining theory and hypothalamic pharmacodynamic indices. TNF-α, NF-κB pathways, changes in intestinal flora, and blood and fecal metabolimics. The open-field test was used to measure anxiety. At the beginning of the experiment, each rat was placed in the corner of the field and allowed to explore the maze freely for 5 minutes. A video camera was placed above the center to record their behavior during the test. This result indicated the successful establishment of the rat model of anxiety disorder. The distance, move duration, speed, and zone crossing were significantly decreased, and the rest duration was significantly increased in the groups that received an oral administration of PCWP compared with the model group. The neurobiological mechanism of anxiety is the result of an imbalance in neurotransmitter function, such as dopamine, GABA, and 5-hydroxytryptamine. A significant decrease in 5-HT, DA, and GABA was observed in the model group compared with the control group. PCWP administration significantly increased the level of 5-HT, DA, and GABA compared with those in the model group. The present study reviewed the presence of a systemic inflammatory response characterized by significantly high pro-inflammatory cytokine levels, including TNF-α, interleukin-6, and interleukin-1 beta in rats with chronic sleep deprivation. Serum TNF-α, interleukin-6, and interleukin-1 beta levels significantly decreased after PCWP administration. Western blotting was performed to evaluate the anxiety alleviating effect of PCWP through the TNF-α NF-κB signaling pathway. A significant decrease of TNF-α, TNF receptor 1, TRADD, phosphorylated IKK-alpha and beta, phosphorylated IKB-alpha, and NF-κB P65 proteins expression was observed after PCWP administration compared with that in the model group. Gut microbiota analysis was performed next. Principal coordinate analysis was performed based on beta diversity analysis to compare the species diversity among different samples. The PCWP group were completely separated from the model group and closer to the control group. The sparse curve flattened out, indicating a reasonable number of samples sequenced. The Venn diagram showed 879 shared OTU among the three groups. Simpson and Channel index were used to reflect the diversity of sample population, and ACE indexes was used to evaluate the richness of the intestinal flora. Anxiety model group shows the decreased diversity and richness of the intestinal flora compared with the control group, and the PCWP group were significant higher than those of the model group. PCWP significantly upregulated the abundance of bacteroides and decreased the abundance of firmicutes. There has a significant increase of pro-vitilice group and ricnolice group after PCWP administration compared with that in the model group, and a significant decrease of Fusicatinobacter was observed compared with that in the model group. Fecal metabolites were measured using an untargeted metabolimics approach. 38 PCWP-related metabolites and 4 metabolic pathways, such as sphingolipid metabolism, taurine and hypotaurine metabolism, vitamin B6 metabolism, and growth cell phospholipid metabolism, were explored. Some studies have shown that sphingolipid metabolism and related enzyme plays an important role in the development of neural disorders and may be one of the mechanisms of depression and anxiety. The results of serum metabolomics showed that 26 biomarkers were significantly changed after PCWP intervention compared with the model group. The regulatory effect of metabolic pathway enrichment on sphingolipid, phenylalanine, and taurine and hypotaurine metabolism, and validation results that showed that PCWP intervention regulated activity of enzyme involved in the above metabolic pathways. PCWP intervention ameliorated anxiety-like behaviors, increased the level of 5-HT, GABA in the hypothalamus. The anti-anxiety activity of PCWP was likely attributed to the inhibition of the TNF-α NF-κB signaling pathway and amelioration of the gut microbiota dysbiosis in the rat model of anxiety. Our findings reveal that sphingolipid metabolism, taurine and hypotaurine metabolism, glycerolphospholipid metabolism, vitamin B6 metabolism, and phenylalanine metabolism were the key metabolic pathways. That's all. Thanks for your listening.